Good afternoon, everyone. It's noon, so I'll start with some opening remarks, and we'll kind of watch as people come in. Um, Welcome, everybody. Welcome to the March 2023 edition of the Ohio River Basin Discussion Series. The topic for today is student voices. We'll be hearing from 3 student presentations today, and I look forward to introducing your presenters in just a few minutes after some opening notes. This series of webinars is organized by the Ohio River Basin Alliance, otherwise known as ORBA, and hosted by Orsanco, the Ohio River Valley Water Sanitation Commission. The sponsors for today's webinar are the Ohio River Basin Alliance, Orsanco, the Foundation for Ohio River Education, otherwise known as FOR, the Voinovich School of Leadership and Public Service at Ohio University, Thomas More University, West Virginia Rivers, the Kenton County Soil and Water Conservation District, and Ohio River Way. I'll be serving as moderator today. My name is Chuck Somerville. I'm Dean Emeritus of the College of Science at Marshall University and the Executive Director of the Ohio River Basin Consortium for Research and Education, otherwise known as ORB-CRE. Before introducing your presenters for today, I just want to take a moment to specifically thank the folks who have done the work of organizing this discussion series and today's event. Jennifer Bowman, whose contact information you see in the chat, is the Director of the Environmental Programs in the Voinovich School of Leadership and Public Service at Ohio University. Jen is also the leader of the Knowledge to Inform Decisions, or the KID, working group for ORBA. Dr. Chris Lorenz is Professor of Biological Sciences at Thomas More University, and Chris also serves as the Vice Chair of the ORBA Steering Committee. Nicholas Callahan is the Environmental Education and Outreach Coordinator at Orsanco. And last but certainly not least, Adam Scott is in charge of the tech for today. He's the Computer Systems Administrator at Orsanco. He makes this all go. Finally, if you're not aware or not familiar with ORBA, it's a volunteer organization that started in 2009 to serve as a forum for discussion and action on water resource issues in the Ohio River Basin. My organization, ORB-CRE, was founded in 1985, and it serves as a forum for academic research related to water resources. We'll keep you updated on ORBA and ORB-CRE meetings that are coming up in the fall. So without any further delay, their questions, if they come in a little during the talk, otherwise we'll have we'll reserve some time for questions at the end of the talk. Please type your questions into the chat, and then I'll relay them to the speakers. So we've got 3 students, as I mentioned earlier. We're going to start with Jonathan Frommeyer, who's a student at the University of Louisville, and freshwater mussels using the Muckett actinonice ligamentina. Jonathan, floor is yours. Jonathan, I think we've got you muted. If you can unmute, get you started there.
Can you hear me now?
Yes, we can.
And can you see my PowerPoint just before I get started, just to make sure?
Yes, we can see it, and we can
Okay,
hear
cool.
you.
Sorry, I
Thank
can't see the chat with it up. Thank you. So yes, my name's Jonathan Frommeyer. I'm a PhD student at the University of Louisville. My research involves actinonice ligamentina and working with those in the Kentucky River and Salt River systems. I'm do most of my work in conjunction with Kentucky Fish and Wildlife at the Center for Mollusk Conservation with Dr. Monty McGregor's and his crew. So that's where I do most of my research. So to start off, a lot of my research involves population structure and correlation with the mussels and their fish hosts. So mussels have specific fish hosts that they use for continuing their life cycle, which I know Sawyer will be talking about a lot today. And what I'm going to be talking about most of through this presentation is actually looking at the sampling for mussels and the fish in the different sites that I will be using, as well as I'll be doing a comprehensive study of fish hosts for Actinias ligamentina, which is the mucket, as well as Elliptiodilatata, which is the spike. And field sampling, this is actually one of my sites that will be conducted. So they're usually smaller rivers that can actually be able to be waded through. And mussel collections for each of the sites in these rivers have abundant mucket population, and we'll use a quantitative mussel sampling. For this, it'll be usually a 5% of the total area in the site for the adequate mussel sampling. And fish collections for these will be done via backpack electrofishing, as well as seine nets. So we'll mark off a lot of the section of the river, the exact place where we'll be sampling mussels, which looks something like this. And so it'll be an area blocked off with about 20 meters north and south of the river, upstream to downstream, and then the length of the river left to right from bank to bank. And these are done using snorkeling for mussel sampling, and like I said, backpack electrofishing using nets, which is a very effective way of sampling everything that will be in that stretch of the river. And so the quadrat sampling looks something like this. Like I said, upstream, downstream is about 20 meters. We'll define the boundaries specifically and then create what are called lanes within this. So that way we can make sure that our sampling is randomized. Because like I said, we want at least 5% coverage. Typically we will do closer to 10% just to make sure that we're covering everything, 5, 7, or 10, just kind of depending on the river. What's blocking it, like you can see in this example that I have here, if there's a big tree in the way, something like that, it makes it kind of difficult sometimes to sample. But everyone that's sampling will be using snorkels and dive gear just to make sure that obviously you're safe, but it's the most efficient way of sampling for these mussels. And so this is a better picture. If we need it, sometimes we'll use a hookah rig like you can see in the picture on the right. And each of the markers are marked with a transect. And like I said, we'll have specific lanes to set up. And typically they're lanes of 3 meters by 20 meters long. And so in this picture you see here, each lane will be split into 3 meters, and that is to have even more randomization within our grid, so we're not overlapping as much and kind of congregating in a specific area. So if we randomized the entire area, you could have big clumps of sampling areas, and we just want to avoid that. So it's kind of systematic randomization where we randomize within the 3 meter lane along the entire 20 meter grid. So you can see in the picture on the left, that's a 2x1. We use just 1x1, 1 meter squared sampling quadrats. And in the example here, you'd start in the starting point. So you have M in the middle, L for the left, and R for the right. And for example, there in the starting point, we would be starting at 1M. And so from there, we would use Excel or randomization grid to be able to show which are the ones that we're doing. So for example, if we have a 20 by 20 grid, we're going to do about 20 samples at minimum to make sure that we're covering the 5% that we need for sampling for within our quadrats. So the big question that I'll be asking with this is there a correlation between the muckets population and its fish hosts? So like I said, I'll be doing a quantitative sampling for these mussels as well as fish, and then doing a comprehensive fish host study. So we'll know the fish hosts, be able to determine the populations of these mussels and fish that are present in the area and see if there's any kind of correlation, and then be able to use this and apply for other species too. So we'll also be observing the spike, Elliptiodilatata, like I said, to be able to go through. And then these are two very common species. That's why they were chosen. They have two different brooding styles, Actinias being long-term and Elliptiodilatata being short-term. And to be able to look at, is there a possibility of seeing, okay, muckets are in high population, spikes are in high population, their host you would think would be in high population if they're using one, or if they're broad host users, or broad users of hosts, which some mussels are, then you would assume that a lot of the hosts that they worked really well with in the lab would work and have high population numbers in the wild. And uh, like I said, the sites will be broken off just like I showed you before. They usually encapsulate a riffle and a run. It's the best way of doing it. The pools are a little harder to sample, plus there's not as much mussel habitat there, so we want to be and have the best possibility of sampling these in their habitats, as well as keeping it as random as we can to not lock in on an area. And like I said, there is a fish host study as a part of it, comprehensive for both of these species. I know Sawyer's going to talk a lot about that. I just wanted to show a couple of these images and hit home again that mussels have a specific life cycle where they rely on the fish to be able to continue. And you can see their larvae are called Glaucidia. Some species have lures. That's what Glaucidia looks like in the upper right. And then they actually attach and are ectoparasites on the gills of the fish. And for about 2 to 3 weeks, then they will be able to fall off. And so for part of my study too, looking specifically at the females in the Glaucidia, getting fecundity estimates for the muckets and the spikes to determine what's the possibility of population in certain areas, and also to observe the Glaucidia of these common species that has minimal research done. So we want to do further research, look at the Glaucidia, what makes them special, and kind of unique to each of the species. How do they attach to their host? For more details like that. And you can see on the right, the gills have their gills just like marsupial pouches where they hold the Glaucidia, and each of those gill slits would hold Glaucidia, thousands of them. Sometimes female mussels can have up to hundreds of thousands of mussels or of Glaucidia, excuse me, to be able to put onto a fish. So that's also something to look at too, is would a broad user of hosts like a mucket have more Glaucidia with inside of them because they're more broadcast spawners than something that would use a lure? That's another thing that we're going to be looking at here. And then Glaucidial extraction with these mussels are done by needles, which I know Sawyer will talk about more about. I was just showing an example here, and that's actually a plain pocketbook that we're looking at and pulling out the Glaucidia of there. And Dr. McGregor, excuse me, is there with me doing that, and that's what we'll do for a lot of the study. And so the overall goal of my project is to understand the mucket's life history. It's a common species, and to be able to study aspects for other rare, threatened, and endangered species. So hopefully the knowledge that I'll be acquiring with the muckets, since they're common, we're not harming a lot of the more rare species. We're using something pretty common found all over the state and within the basin too. This can be applied for other species as well. And a lot of mussels, which almost all of them are, declining because of water quality for various reasons, dams not being able to reach their fish hosts, etc. We want to know if this can be used more for conservation as well. So conservation is obviously very important. It's specifically of mussels because they're very important for freshwater ecosystems. A lot of times they're ecosystem engineers. They are very important for the nutrient cycling that goes on within these systems as well. And could it pertain to other species, more rare species as well? If we know that there is a correlation between fish and mussel species when it comes to releasing mussel species to certain areas, which Fish and Wildlife does a lot, is it important to know what fish fauna is there too, which I like to think that it is. It's very important to make sure that their fish hosts are there, obviously, so we continue recruitment in those specific areas. And so that's something that's important to me and why I'm focusing on a lot of this research. And so that's kind of the end of my talk there. This is the Green River site that I go with. Monte Alata is not my specific site for this, but you can see another example of where fish sampling is done, and fish and mussel sampling is done, excuse me, in this area, and is home to a lot of endangered species. So thank you. If you have any questions, I will take them, or if we're doing questions at the end, that works too.
Thank you, Jonathan. We're within Jonathan's time, so if anybody wants to type a question into the chat, please feel free to do that. We can also have some time for questions at the end if no one has a question ready just yet. So we'll take a minute or two to see if any questions come in. Jonathan, what are your plans following graduation?
That's a good question. Kind of leaving it open, more opportunistic. I really like field stations and um, environmental education, so that's something that I would continue with ideally. So whether I'm working for a university or doing something like Fish and Wildlife, I would be able to do outreach, interpretation at different areas within education, mentoring students, stuff like that.
Great, thank you. There is a question in the chat from Dr. Chris Lawrence. What are some of the fish hosts for spike, if they are known?
So darters are what is known specifically with spikes. Other than that, there's not a whole lot of research out there. Typically, more of like the darter species for sure. Thank you.
Yeah, thanks very much, Jonathan. I enjoyed that very much. So we're gonna go on to Sawyer Lawrence. Please remember that if you've got questions for Jonathan, go ahead and put them in the chat. We'll pick them up at the end of the talks. Our next speaker is Sawyer Lawrence from Thomas More University, and Sawyer is going to speak about fish host analysis of the paper pond shell, Utterbachia imbecilis. Thanks.
Okay, can everyone see everything okay and hear me okay?
Yep, you're good.
Okay, cool. So my name is Sawyer Lorenz. I am a soon-to-be graduate of Thomas More University, and I'll be presenting my research that I did here at the Thomas More University Biology Field Station on the paper pond shell mussel Utterbachia imbecilis. So freshwater mussels have a unique life cycle that Jonathan kind of touched on. It requires a fish host for their larvae, Glaucidia. Mussels of the family Uniatidae specifically typically have this lure-like structure that you can see in figure 2. And it mimics a small minnow or prey species that can draw a larger fish near. And then once the fish gets drawn near, the females will release their Glaucidia, and the Glaucidia attach onto the fish's gills. So you can kind of see this diagram illustrated in figure 1 of the life cycle. Once the Glaucidia attach to the gills, they acquire essential nutrients from the fish, and after about a couple weeks, it depends on what type of mussel it is, but they will fall back off as juvenile mussels. Each mussel species requires a specific fish host species to host their I don't know why it keeps changing on me. Oh, there you go. So each mussel species requires a specific fish host to host their Glaucidia. So not every mussel species can have every fish as its host. It's very specific. This life cycle can be replicated in a lab by artificially infesting fish with Glaucidia from a gravid female mussel. And then artificially recreating this process in a lab can help bolster populations by raising juveniles in a lab and then releasing them into the wild, as it'll increase their survival chances, as that first couple weeks is very essential. And this is important because freshwater mussels are one of the most threatened species due to multiple reasons, but the top 3 would be overharvesting, habitat modification, and invasive species. And then this is just a little bit about the paper pond shell, which is the mussel that I did for my project. They have a very thin and elongated shell. They're typically found in rivers, lakes, and ponds with soft mud or sand substrates. You can find them around here. They are filter feeders like most mussels, primarily feeding on phytoplankton. And their native range is the majority of the mid and eastern US. So they can be found many places. They're actually pretty common for mussels. And so to go about this project, we first reached out to Dr. Monty McGregor at Kentucky Fish and Wildlife. He was very generous and allowed us to get 3 gravid female paper pond shell mussels to bring back to the field station. We then got 4 individual fish for 3 different fish species within the Lepomis family from Jones's. So there's little literature on the known fish host for the paper pond shell, but what we did find confirmed bluegill as a host, so we wanted to go within that family. So we had 4 speckled belly sunfish, 4 bluegill, and 4 hybrid bluegill. And then we brought them back and acclimated them to their own individual tanks. As you can see in picture 2, every fish had its own tank. When we were ready to start the infesting process, we used a large hypodermic needle to inject the water into the gills of the female mussels to allow the Glaucidia to flush into a concentrated sample in a net, like you can see in the 3rd image. That's what I'm doing there. At the same time, one of us used MS-222 to inoculate the fish. This did not harm them. It just simply knocked them out for around a minute to make the infesting process a lot easier. So one of us held the operculum of the fish back while the other flushed the gills of the fish with the concentrated Glaucidia sample, which was 0.25 milliliters, which was equivalent to about 437 individual Glaucidia in that quarter milliliter. We then monitored the tanks and siphoned for juveniles into a small filter. We would look at the collected matter every day under a microscope and count juvenile mussels. Like you can see in picture 5, that's what we would see under a microscope. And we did this every day until there was no mussels found for 3 days in a row. And then onto my results. My results for this study were pretty simple. We were just looking for a yes or no on attachment, and then we also counted the total number of juveniles per fish. So all 12 of the fish produced juvenile paper poncho mussels, so we were really excited. This confirmed all 3 fish species as hosts for the paper poncho mussel. The bluegill hybrid was the most successful of the 3, as they had a total of 83 juveniles fall off between the 4 individuals. Bluegill produced a total of 73, and then the speckled bellies produced a total of 63 juvenile mussels. So all 3 are definitely viable options. And then looking at the results, we can see that all the fish used in the experiment dropped at least 2 juvenile mussels. The average number of live juvenile mussels dropped per fish was about 18.4. The hybrid bluegill had the greatest output when compared to the other 2. Although there was one outlier, one of the hybrid bluegill only dropped 2 juveniles over a period of 1 day, while all the others had a minimum of 9 juveniles over about 3 to 5 days. But despite this, the hybrid was still the most efficient. But again, all 3 were viable options. And you can see in this little clip here, just a video of what we would see under the microscope of a little juvenile mussel moving around. And then just overall importance of this study, by identifying fish host requirements of freshwater mussels, we can help lay the foundation for conservation by providing efficient reproduction methods such as the bluegill hybrid to help bolster populations in a lab and then release into the wild. And then I just want to give a special thanks to all those at Kentucky Department of Fish and Wildlife for donating the gravid mussels, Joneses for donating the fish, and Jonathan Fromeyer and Dr. Monty McGregor for all their contributions and help with the logistics of the project.
Thanks very much, Sawyer. That's a very interesting study. We're well within your time. If anybody has any questions to put in the chat, we'll pass those on. If you still have control of the screen, would you mind going back to your figure 2? I wasn't familiar personally with just how realistic those lures were on mussels.
Yeah.
That's pretty amazing.
Yeah, I wanted to put a video on here of one moving, but I didn't know how well that would work with the video, but they move in the water just like a little minnow would.
Yeah, you can certainly see how those would be effective. I was also pretty impressed with the number of juvenile mussels produced in a pretty short time. So it looked like the longest period was around 5 days. Is that right?
Yeah, no. I think around like day 7 we started seeing mussels fall off, and then once they did, it all happened pretty quickly. So it was really exciting to watch.
Yeah, that's very quick. there's a question in the chat from Jen Bowman at OU, and her question is, what is the survival rate of the juvenile mussels?
Jonathan could probably speak a little bit better to that. I know after falling off, the first couple months is pretty essential. I know at Kentucky Fish and Wildlife, they raise a lot in the individual tanks. We tried to keep some going, like these juveniles we collected, and we weren't pretty successful. I think they're very sensitive to water conditions and the nutrients that you're feeding them. So I would say it's pretty hard, but definitely possible. They've got it down at Kentucky Fish and Wildlife.
There's another question coming in from Drew Seiler. It says, since the Glaucidia sap, maybe not a question. Oh, it is a question. Since the Glaucidia sap nutrients from the fish hosts, is there a certain threshold where Glaucidia impacts survival of the fish?
Yeah, so it's kind of a highly debated topic of whether or not this is a parasitic relationship. Most biologists are leaning towards yes, it is parasitic, because the fish do build up a tolerance to the Glaucidia attachment. So that's why we got our fish from Jones's, so we could assure that they would have never been infested with any Glaucidia, and they wouldn't have a tolerance to it. So I think, and whenever the Glaucidia or the juveniles fall back off, they do rip through the tissue of the gills a little bit. So it's nothing that was going to kill the fish, but after a while it does start to impact them. And so that's why they build up a tolerance to them.
Yeah, following up a little bit on the earlier question, do the juvenile mussels require a certain type of substrate? I remember that you said that the paper pond shell likes a kind of a muddy habitat. When they're first establishing, do they require any kind of a hard surface to attach to, anything like that?
No, I think it's kind of specific to mussel species, but I know for most of them, you could just put them in a tank with water and maybe a little bit of silt on the bottom. Whereas mussels like zebra mussels that are invasive cling to those hard substrates. So I think it's specific per mussel species.
Great, thanks a lot. There's still time to put questions in the chat. We can, and we'll have some time for questions at the end of all three talks. But if there aren't any additional questions right now, we'll go ahead and move on to Tyler Rowan, the third talk for today. Thanks, Sawyer. Thanks very much. Tyler is a PhD student at the University of Pittsburgh, and his topic is on rainfall thresholds for landslide occurrence in Pittsburgh, PA, using 311 citizen science data. Tyler, the floor is yours.
All right, should be up and running now. Thank you for having me. As he said, my name is Tyler Rowan. I'm a PhD student at the University of Pittsburgh in the Geology and Environmental Science Department. And the focus of my PhD has been on examining natural hazard systems through empirical and machine learning modeling. And today I'm going to be talking to you about rainfall thresholds for landslide occurrence in Pittsburgh, Pennsylvania. So up here on the right, I have a large landslide. It's actually a couple blocks up from the Department of Geology here that we've been working on, and that's been reported through 311, just to kind of give you a good visualization of some of the landslides I'll be talking about throughout this presentation. So now on just to talk about landslide hazard in southwestern Pennsylvania. So according to this U.S. geological map, southwestern Pennsylvania has some of the highest landslide hazard in the nation, as well as most landslide occurrence in the nation. So why is that? What makes southwest Pennsylvania so landslide prone? So while there's many factors that contribute into it, four that really stick out is there's a lot of abandoned coal mines, which can reroute drainage as well as undercut hill slopes. There's a very clay-rich lithology in the area, with the Pittsburgh red beds being notoriously a geotechnical nuisance with slope failures. a very hilly topography being north of Appalachia, which is going to unbalance this driving versus resisting forces and how we like to think and model landslides. And then what I'll be focusing on today is heavy precipitation in southwestern Pennsylvania. We get a lot of rain here. We get a lot of rain every year, and that contributes greatly to landslides that we get here in Pittsburgh. And I know most likely when you think about rain-induced natural hazards, you think of flooding, but also landslide occurrence increases or correlates greatly with large rainfall events. So before we get into the landslide and rainfall thresholds, let's first just discuss how does rainfall induce landslides. So really there's two primary players when we're talking about rainfall induced landslides. Firstly, they can weaken the bonds between the soil particles by dissolving or washing out the materials that kind of hold and cohese the soil together and add to that soil strength. And they can also erode the sediment at the base of the slope. So undercutting that slope, rebalancing that driving versus resisting forces, and then causing a slope failure. So knowing landslide natural hazards are such an issue, what are some tools that we have in our arsenal to try to mitigate landslide damage? And thinking about this in both a spatial and temporal aspect, spatially, you can do landslide susceptibility maps where you're looking at a certain location based on past locations of landslides as well as topographic factors to calculate probability across a designated study area. And then thinking about this on a temporal aspect, we have early warning systems like the one I'll be talking about today, which are rainfall intensity duration curves, where we can look at past storms that have induced landslides and create a threshold based on the past knowledge or that we've gained from these previous landslide events. So a main thing that's important to creation of either of those mitigation tools is a robust landslide inventory. And for the landslide inventory that we're going to be using in this study, it's actually based on the 311 system. Don't be worried if you didn't hear about it at all before. I actually just learned about it going through the study, but what the 311 system is it's a non-emergency phone number that allows citizens to report a variety of different issues to municipal services to be fixed, with landslides being one of them. The system is in place in over 300 different cities in the United States and Canada. And from this system, we can create a very robust landslide inventory for the city of Pittsburgh. So why use 311? What are the potential benefits of it? So the potential benefits of using a 311 inventory is, firstly, it's publicly available. If you want the data, you can go onto their website right now and download it. It updates in real time. So as citizens report these issues, they get added to the massive 311 spreadsheet. And as I said before, importantly, it records both location and time of the events. So we get both the location and the reported time of these landslide events. It's a pretty low cost and low effort process when you're comparing to making a landslide inventory based on remote sensing or field work. So really you can gather a lot of data quickly and at minimal cost. However, it's not all roses. There's definitely some issues with it. Because we're dealing with a citizen science reporting system, there can be significant inaccuracy. You can get error in the reported location from the citizen. They can report the street intersection instead of the actual location of the landslide. And sometimes you'll get multiple reports on the same landslide event. So you get probably multiple citizens just seeing and reporting the same landslide. So because of this, we did some field validation of these 311 landslide events in the spring of 2019. Here are some pictures of landslides we visited with fellow graduate student Nick Wondolowski there for scale. And from this field validation effort, we found that out of the 77 sites we were able to visit, 55 of them actually contained evidence of a landslide being there. Out of the 22 false reports, 7 of those 22 false reports were actually duplicate reports of the same landslide. And then the mean distance away from the reported location, so where the landslide was actually reported versus where the landslide actually was around 104 meters displacement. And here's a map of where we validated the true and false reports, as well as the unvalidated landslide reports that came in after that process was completed. So now on to discussing, what is rainfall intensity duration curve and how do we use it to be a tool against mitigating landslide damage. So because the 311 data includes both location and time, we can correlate these landslide events with past precipitation data and plot them on a log intensity and log duration plot. And from these events, we can actually create an exceedance threshold based on the linear regression of the points. And commonly you do about a 5% exceedance threshold. So what information do you get from this? So the information you get from this is that if we have a storm event and that storm event falls above the calculated threshold for the region, we would suspect that that storm event would be associated with landslide events. Contrasting, you have a storm event that falls below your calculated rainfall threshold. You would expect that event to not be correlated with landslide events in the specific study area that the rainfall threshold was calculated for. So looking at our 311 precipitation data, this is the 311 precipitation data on an intensity duration plot. Basically, we combine the reported time of these landslides with hourly rainfall data from the nearest NOAA monitored rain gauge system. So out of the 1,008 311 reported landslides from 2015 to 2022, about 52% were associated with a rain event. So one thing, for those of you who don't look at these regularly, that's kind of unique about this, or an outlier, is we have all these points down here at the bottom left-hand corner of the plot at these very low duration, low intensity events. So trying to tease out why we're seeing this in our data, we went back to our field validation efforts. So overlaying now are where we saw our false landslide reports versus our true landslide reports. And we start to see a similar pattern where a lot of these false landslide reports are the reports that are making up the bottom left-hand corner of the reports we see. So in order to kind of shore up some of the accuracy of this, from these false 311 reports, a 0.19 millimeter threshold for rainfall intensity was used for our exceedance threshold calculations. And again, here is the 5% exceedance threshold for our 311 landslide reports. Because we're dealing with this uncertainty with citizen science, it's important to also calculate the uncertainty in this rainfall threshold. So using a Monte Carlo procedure, with 100 different iterations. In each iteration, we took 100 different randomly selected samples of these landslide events, calculating new alpha and beta values, which kind of define the shaded area which makes up our uncertainty range for our rainfall threshold using this 311 data. Another way to validate this data is to look at previous work that has also been done in this area, different methodology and using much smaller sample size. We see Ashland 21, who was looking at larger storm events, specifically after Sandy, and his threshold has a little bit of a higher slope, likely biased due to some of those larger rain events he was looking at. We have PennDOT District 10 data, which actually follows very closely and within the uncertainty of the 311 plotted threshold. And then I also added Kane's global threshold here, just for those of you who know the material and are interested in seeing how this aligns with the global threshold. The differences here are likely due to local and regional controls on landslide versus looking at it in a global context. So another issue we have here in Pittsburgh, Pennsylvania is landslide reactivation. So these are two images of the exact same location 2 years apart. The left one is during a 311 field validation, and the right one is during revisiting the site during another field exploration. As you can see, the small landslide reactivated and now became a much, much larger landslide. And this reactivation occurs pretty commonly in the southwestern Pennsylvania area. So when we're looking for reactivation landslides and trying to explore the relationship between this and rainfall metrics, we actually went back to an old USGS landslide map from the 1970s and 1980s, where they mapped old inactive landslide deposits. So 311 reported locations that fell within these old landslide deposits were considered reactivated landslides. So then we now had an inventory of both reactivated landslides as well as our non-reactivated landslides and computed thresholds based on those data points. what we see, as shown in the graph over here to the left, is that the reactivated landslides are occurring at a much lower rainfall intensity duration. So it takes a lot less push to fail with these reactivated slopes than the non-reactivated landslides. So less rain is required to really get these things moving down slope. These intensity duration plots are also a great tool for exploring the relationship between other factors and rainfall metrics, such as slope here, looking at slopes broken up into different brackets of 15 degrees. We see that slopes that occur at lowest degrees require the most rain input, so the largest intensity or the larger duration storms, and this was just extracted from slope or other topographic maps at the reported location of the 311 landslide. We can also look at things such as distance to the nearest stream or vegetation cover. With distance nearest stream, we're seeing our landslides occur for our very close streams, our 0 to 50 meter streams, at our very long duration storm events. And this likely could go back to washing away the toe of the slope, undercutting the slope when we get high flows in those streams after long duration storms, and then that undercutting of the slope likely leads to the slope failure. With vegetation cover, a little bit less of a clear picture, but we do see that heavily vegetated areas require more rain input for failure than what we're seeing with loosely vegetated or impermeable areas, likely due to the cohesion that the vegetation adds to the soil, as well as the saturation that the vegetation can soak up, helping desaturate those hill slopes. So thank you for allowing me to give you a quick glance into the use of citizen science for landslide mitigation. So far, these are the two products that we've been able to produce using the 311 citizen science data, with our rainfall threshold over here to the left and our landslide susceptibility map used in a previous study over here to the right. So now I'll just add some of our conclusions up there, and I'll take any questions you guys have. Thank you.
Thanks, Tyler. Very interesting study. I can certainly see some application of your work there. On your slide 18, thought it was pretty interesting that there's a that high susceptibility region in Pittsburgh there. Looks like Mount Washington. Is that right?
Oh yeah, Mount Washington is definitely an area where we see lots of landslides.
Yeah.
Mm-hmm.
So have you been able to work with the city at all? Have they shown interest in your kind of prediction susceptibility?
So luckily, when I started this study, when I first reached out to them, they were not really willing to share data. But funny enough, the iRise consortium, actually run through the engineering department, has now partnered with PennDOT, and we're actually working with them towards modernizing their landslide database, as well as working towards these issues. So 2018 was really a bad year for landslides in Pittsburgh, Pennsylvania. So the money that the repairs cost kind of pushed the initiative. And yeah, definitely a big thing. If you're interested, look up iRise. They're doing a lot of great different projects involving this field.
Yeah, very interesting. There are a couple of questions in the chat for you. Let's see. The first one is from John Hageman, Thomas More University. Hey, John, how you doing? And the question is, which vegetation species are best to prevent or reduce landslides, and which would be the worst, if you have that information?
Yeah. So I don't have specific species, but maybe I can just talk into a little bit of like how vegetation is going to stabilize a hill slope. So when we're dealing with larger, denser species of vegetation, where we get a lot of root cohesion within our soil, and we're really stabilizing that hill slope, we generally have a more stable hill slope. Or if we get kind of these less dense populated species and more kind of widely spread out, you don't get as much stability with the hill slope there. I believe there was actually a good study actually put up by somebody before me looking at boreal trees and their influence on slope stability. So it's definitely something that's been looked at different species being influential in slope stability for sure.
Thank you.
Yeah, we've got a second question also from Thomas More University. This is Dr. Lorenz. Very interesting study. How might climate change impact the frequency of landslides?
Yeah, that's a big question and a big motivating factor, because we expect with climate change, right, we'll get more intense storms more frequently. So with those more intense storms, we would expect to have more landslides. So as we get into a climate that has more extreme events, specifically more extreme precipitation events, I would expect there to be more landslides and it be a bigger problem than what we have today. So definitely something that could grow in the future.
And we've got a question from Jen Bowman at Ohio University. In general, do you see streams in Western PA have a higher level of turbidity, TDS and TSS, because of the increase in landslides?
Great question, but I haven't looked into any turbidity data, unfortunately, regarding the landslides. So I unfortunately do not have an answer to that. Great question, though.
Yeah, I had that similar thought when you showed your earlier map of the regions that are susceptible to landslides. It kind of follows the Appalachians, and a lot of that covers the Ohio River Basin.
Yeah.
There's a new question in the chat. I think slide 16 indicated that the smaller slope required more intense rainfall, but the steepest also seemed to require more intense rainfall. Do the steeper slopes tend to have more vegetation? Is a possible explanation for the higher rainfall?
So that could be one. It could be a couple of other factors. That last bracket of 45 degree plus slopes has the smallest point. So it could just be some data bias I'm getting in there. But it could also be a bunch of different factors, such as, we could have different aspects that are controlling the evapotranspiration on those slopes based on the, and as well as, like you were saying, different high degree slopes could be different species and leading to different stabilization of those slopes. So definitely a couple of different factors that could be the reason for that. But
yeah, thanks. I want to remind the audience that if you've got any questions for Jonathan or Sawyer, we can also entertain them at this time. And any other questions for Tyler? I had one follow-up on the 311 system. Do you know who funds that system?
So the person who's in charge of it right now is Allegheny County Emergency Services, is really who's in charge of it, with the Department of Public Works really servicing a lot of the requests. But I do not specifically know where the funding comes from. I'm guessing because it's limited to the city limits of Pittsburgh, it's probably funded through one of their sources, because I'm guessing if it would come from Allegheny County or the PennDOT districts, it would have better coverage. But because it's actually limited to the city limits, I'm guessing it's from a city funding source.
Yeah. I was impressed with the number of different informational points that are reported through 311, and your idea to use that information is impressive. And it seems to me like it could be used for those data could be mined for other studies like yours as well. There's another question in the chat from Dr. Hageman at Thomas More. North-facing slopes versus south-facing slopes. Have you seen any difference there?
Yeah, so actually that goes into when we were doing our landslide susceptibility mapping. We actually look at all the different landslide related factors and rank them based on influential factors. And sometimes we'll do things where we'll break them down in categories. And we actually did see that north-facing slopes do have a larger influence on landslide occurrence. It's actually north-northeast were the largest two brackets. And this could likely be due to actually evapotranspiration, where with the setting sun, funny enough, as the best explanation we had for that. So yes, we did see north and northeast facing slopes have a better correlation with landslides than other aspects.
And we've got a question from Dr. Lazorchak, Dr. Jim Lazorchak, EPA Cincinnati. Why do you call the data collection citizen science? The data is not being generated in collaboration with professional scientists. Correct. Is that correct?
Yeah, so it's the only collaborators are the citizens that are reporting it and the municipal officials. So I may be, although it has been defined as citizen science before, but that's just, yeah. So it's citizen reported to the municipal officials, essentially, this part of the frameworks.
Yeah, I noticed that when you were talking about it, one of the issues that came up was duplicate reports of a single landslide. So there's, as far as you know, is there anybody that curates that information in the database, or is it simply kind of collected as reported?
Yeah, so I wish it was curated. That would make the field validation a little bit better, but unfortunately, once it's reported, the report is what they, at least the publicly information. I'm sure maybe when the City of Public Works goes and visit the site, they probably make some remark, right, if there's nothing there for them to do. But from the publicly available data that I had access to, that information was not in there, unfortunately. I wish it was. That would have been great, especially to deal with some of the validation and dealing with the uncertainty of the data.
Thanks very much. So we've got about 9 minutes, and I'm happy to entertain any other questions for not only for Tyler, but for Jonathan or Sawyer. As I keep an eye on the chat, I just want to say to each of you, thanks very much for presenting today. As a person who has been working in the basin for a long time, it's both encouraging and enjoyable to hear from students working in the basin. I really appreciate your willingness to present and the quality of your work. Comment in the chat from Dr. Lorenz. Thanks to all the presenters. Keep up the good work, and hope to see you at ORBA meeting this fall. Keep an eye on information for those meetings that'll come up in the fall. Usually they're in October or sometimes in early November. We'll get those dates out to you as soon as we have them. As we're coming up on time, anything from comment from Jonathan? Thank you for having us. Appreciate the invite. Very well conducted. Thanks, Jonathan. Appreciate that. Jen or Chris or anybody, any comments or notes for the good of the order for the attendees today?
This is Jen. Thank you, Chuck. I would say this concludes our winter webinar series, and we will be working as a committee over the summer to solicit new topics and speakers for a probably a late fall, early winter series to kind of give our fall time period a break so that we can attend the ORBA and ORBCRE conferences. So please reach out if you have some topics or issues or ideas for future webinar discussion series. Thank you.
Yeah, thanks, Jen, and thanks for all your work on this over the course of the series. It's been really great. We still have a little time if there are any questions. If not, feel free to go off the rest of your day. I want to thank the students for presenting today. Thanks to Jonathan, Tyler, and Sawyer for your presentations and for your work. Keep in touch with us as you go forward, because having you folks in the basin has been a great thing, and want to make sure that if possible we can keep you working here. Comment from Laura: student presentations were great, thank you. Comment from John Hageman: thank you to all involved in setting these up. And a thank you from Heather Strauss. Thanks, everybody. I think if there are no more comments in the chat, we can wrap up for today. Thanks again to ORBA and ORBCRE and all the sponsors for today's discussion series. Thanks, everyone.
Thanks. Thanks, Chuck. Bye-bye.